with undergrad, Dr. Jessica Hua. And Dr. Hua has received her PhD in Ecology and Evolution from the University of Pittsburgh in 2014 and was a postdoctoral scholar with the Department of Forestry and Natural Resources at Purdue University from 2014 to 2015. She is currently a professor in the Department of Biology at Binghamton University, in addition to being the director of the Center for Integrated Watershed Studies at Binghamton University. She has most recently received an NSF Career Award in Environmental Biology. Dr. Hua's lab focuses on understanding the mechanisms shaping ecological interactions, evolutionary processes, and eco-evo feedbacks, with emphasis placed on human disturbances such as pollution, temperature shifts, and invasive species. And I know she's going to talk a little bit more about her lab's work in this presentation. So I will hand it off to you, Dr. Hua.
First, I wanted to say thank you so much, Kim, for the invitation. It was such a pleasure to get to catch up, and I really appreciate you all being here today. I am very excited to have this chance to share with you all some of the research that my lab has been doing for the past few years. So I'll start broadly. We all recognize that the world is rapidly changing in many different ways, from things like emerging infectious diseases, changes in our climate, pollutants in our environment, habitats being lost or modified in unique ways. And so under this broader umbrella of global change biology, my lab at Binghamton, we're interested in understanding how environmental change specifically influences host-parasite interactions. And so when it comes to host-parasite interactions, one approach might be to ask, well, what are factors that influence the host's ability to defend itself against parasites? Or alternatively, from the parasite perspective, what are factors that influence the parasite's ability to infect hosts? Now, building upon this traditional model, the field of disease ecology has increasingly recognized the importance of the environment in shaping hosts, parasites, and also their interaction. And so me being trained as an ecotoxicologist, when I'm thinking about the environment, oftentimes I'm thinking about pollutants and how they influence hosts and parasites. And so with this traditional disease ecology triangle, one of the things to keep in mind is that these interactions don't occur in isolation. In other words, in nature, there are often multiple hosts interacting with each other that can influence disease outcome, or there might be other stressors in the environment, like invasive species that interact with pollutants to affect host parasite outcomes. And so this brings me to the first theme of our research lab, and also the first theme of this talk, which is, can we integrate ecological complexity into our understanding of disease outcome? Towards this goal, the first section of this talk, I'll focus on some of our work on secondary salinization. I'll start by asking a simple single host, single parasite question: how does salt influence host parasite interactions? From there, I'll build in a little bit more complexity, asking about how salt influences multi-host interactions. And then from here, I'll transition to the ecosystem level. How might changes in abiotic conditions interact with salt to influence hosts and also their disease outcome? So in addition to integrating this ecological complexity, my lab is also interested in understanding the evolutionary implications of environmental change on host parasite interactions. When it comes to evolutionary responses to pollutants, there are many examples, from things like the evolution of antibiotic resistance, evolution of pesticide tolerance. And so in my lab, we're also interested in applying this evolutionary perspective into this disease triangle framework. So the broad question in the second section that I'll talk about will be, how does selection by pollutants influence host parasite interactions? And to keep it simple, in this section, I've organized the talk based off of work on antibiotics as well as pesticides, and it will be broken down by the different components of that disease triangle. Lastly, in addition to research, we know that another crucial aspect of my lab, given that global change biology is so intimately linked with humans and society, is it's important to me and also members of my lab that we find ways of effectively communicating our research to broader audiences. And so in the last 5 minutes of this talk, I just wanted to provide just a little bit of an overview on some of our work on how art can facilitate science communication. So this is the outline for today, and I will go ahead and jump right into the ecological perspective and talking about how secondary salinization can influence host-parasite interactions. So secondary salinization, the influx of salt into freshwater ecosystems, can occur a variety of different ways, from coastal flooding to the use of road salts during the winter, and also agricultural practices like irrigation. So to understand how sodium chloride influences host-parasite interactions, my lab works on a variety of pathogens and parasites, but for this question, we'll focus on trematodes as the model parasite. This is the free-swimming stage of the trematode Echinostoma trivolvus. It is free-swimming and it infects tadpole larval amphibians. To infect the amphibian, the cercaria in this free-swimming stage will swim up the cloaca of the tadpole and encyst in the kidney. Those are the metasecarial cysts that you're seeing in the image below the tadpole. So, does sodium chloride modify host-parasite interactions? We used wood frogs as the model host, and then trematodes in the free-swimming cercaria stage as our model parasite. Throughout the talk, I'll highlight the student that was responsible for each of the projects. This work was done by my most recently graduated PhD student, Dr. Nicholas Buss. He took wood frog tadpoles and exposed them to 4 different concentrations of sodium chloride. For reference, seawater is about 19.4 grams per liter. The highest concentration detected in runoff was reported at 18 grams per liter of chloride. The highest concentration of chloride detected in wetlands was about 4.6 grams per liter. However, the majority of wetlands sampled range anywhere from 0.1 to 1.1 grams per liter. Here are the concentrations we use. They're environmentally relevant, including that lower concentration, which is found in concentrations that are commonly found in nature. And also these concentrations are sublethal to our wood frogs. This is the lethal concentration 50 at 4.1. So after exposure to these sublethal concentrations, Nick exposed them to 50 of the trematodes, and he counted how many of these trematodes successfully encysted. Moving on to the results, on the x-axis, we have sodium chloride concentration. On the y-axis, average number of cysts. When not exposed to salt, this is what we find compared to when the tadpoles were reared in a sublethal dose of sodium chloride. In all cases, we found that exposure to sublethal concentrations of sodium chloride increased tadpole susceptibility to this very common trematode. And so taking a step back, what are the main take-homes of this study? We think that the first is, in the field of toxicology, there is this emphasis on what are the lethal effects of different pollutants. We think that studies like this underscore the importance of considering these lower but environmentally relevant concentrations, because while they may not be lethal, they can still have negative consequences, for example, by influencing the susceptibility of a host to its parasite. Now, of course, one limitation of this particular study, as I alluded to before, is that it focuses on just a single host and a single parasite. When in nature, there are often multiple hosts interacting with each other as well as with the parasite. In fact, this relationship between host biodiversity and disease is a topic that has been hotly debated in the field of disease ecology. So this idea of the biodiversity disease risk debate. And so in this slide, I'll talk about what is the biodiversity disease relationship and also why people are so interested in it. So when it comes to biodiversity disease, oftentimes we hear about the dilution effect. The idea that as species richness, species diversity increases, you get the overall decrease in disease in a particular community. And so because of the benefits of increasing species diversity, this provides a clear reason for why people might want to protect species diversity. You get the benefit of reducing disease. The challenge, however, is that sometimes in the literature there is a dilution effect. Sometimes there's no relationship, and other times we get the opposite relationship, where as you increase in species diversity, you actually get an increase in overall infection. And so in this section, what I'd like to do is talk about one factor that we think might be contributing to the equivocal nature of this biodiversity disease relationship. Before I do this, I'm going to take a step back and just talk a little bit about the mechanisms that we think, or theory thinks, is driving the dilution effect. To do this, I've created two hypothetical communities, a lower host diversity community with just the blue species tadpole, and then the higher host diversity community with 3 species here, blue, red, and yellow. So in this hypothetical example, the lower host diversity species are highly susceptible hosts. In other words, you have parasites in the water. If they are to find the host, they're going to be highly successful in infecting these hosts. In contrast, theory predicts that in communities with higher host diversity, there's a higher likelihood that you might have hosts that are less susceptible to the parasite. So in this scenario, you have parasites in the water. These less susceptible hosts can act as decoys. They'll draw the parasite towards them, and because they're less susceptible, they're less competent hosts, these parasites don't successfully encyst. And so in this scenario, you see that as you increase in diversity, you get a reduction in disease. So graphically, it would look something like this. Now, one challenge with these types of studies is that they often occur in benign environments. And this is important because as I just showed, in the presence of a pollutant like sodium chloride, patterns of susceptibility can change. In the research I just showed you, tadpoles became more susceptible to parasites. And so in this situation, in the presence of a pollutant like sodium chloride, we predicted that if it changes susceptibility to hosts, then that benefit of increasing host diversity no longer stands. Perhaps the red and yellow tadpole are no longer your decoy hosts in the presence of salt. So you might get something that looks like this. And so we predicted that pollutants can influence the magnitude of the biodiversity disease relationship by affecting patterns of susceptibility to the parasite. To address this prediction, there are 2 questions we had to ask. First, we had to know, what does sodium chloride do to modify host susceptibility to parasites? From there, once we know what salt can do, we can then make predictions about how salt contamination can modify that magnitude of the biodiversity disease relationship. Again, this is work done by Dr. Nick Buss. So to answer that first question, Nick repeated the methodology from the first experiment, except he just expanded it to multiple species. He exposed them to sublethal concentrations of salt and exposed them to 50 trematodes and counted however many successfully encysted. On this data side, on the x-axis, we have the different species. On the y-axis, again, the average number of cysts per community. The solid bars here represent communities that were not exposed to salt. What we can see here is that there's no difference in susceptibility to parasites across these 3 different species. However, in the presence of salt, we find that peepers became more susceptible to the trematodes. Wood frogs also became more susceptible to the trematodes, and toads did not change in susceptibility. So now that we have an understanding of what sodium chloride does to host susceptibility, we can turn and make predictions about how this salt contamination can modify the magnitude of the biodiversity disease relationship. So I'll start first in the absence of salt. All 3 species have similar susceptibilities to the parasite, and so there isn't any one species that might be acting as that decoy. So increasing species diversity isn't going to, we predict, have any benefit on overall infection. In contrast, in the presence of salt, we found that toads are significantly less susceptible to the parasite. So in this scenario, we would predict that as you increase in species diversity, there's an increased chance that you have toads in your community, and therefore there would be a benefit of increased species diversity and on overall infection. So we predict that in the presence of salt, because there's variation in susceptibility across species, we would see a dilution effect. But in the absence of salt, we would not. So to test this, this question, we generated several different types of communities. We had single species communities with just peepers, wood frogs, toads, 2 species communities, and also a 3 species community. So the one thing to note here is while we manipulated the number of species, we kept the number of individuals consistent throughout. We exposed them to salt or no salt environments, added trematodes, and counted how many of those trematodes successfully encysted. Okay, so on this slide, I'm showing data all from the communities that were not exposed to salt. To orient everyone to the x-axis, we have, as we go from left to right, lower host diversity to higher host diversity. On the y-axis is cis per community. In the first panel, I'm showing all of the different communities compared to just peeper communities, all the different communities compared to just wood frog, and then just toads here in the last panel. Again, the prediction is that when we're comparing across these different species, they all have similar levels of susceptibility. So we predicted no benefit of increasing host diversity, no dilution effect. And I won't spend too much time on this slide, other than to show that we indeed found no evidence of a dilution effect in any of the comparisons here. So, in the absence of sodium chloride, we found no dilution effect. Transitioning to an environment with salt. As a reminder, salt made peepers and wood frogs more susceptible to parasites, and importantly, toads were less susceptible to parasites compared to these other 2 species, potentially acting as that decoy. So we know that pollutants modify patterns of host susceptibility. Does this change the biodiversity disease relationship? On this figure, we predicted peepers and wood frogs were highly susceptible in the presence of salt. And we know that toads are less susceptible. So we predicted that any community with a toad would see a reduction in overall infection. For the most diverse community, we also predicted a decrease in infection. But in this community, because there were only 2 decoy species compared to the 4 susceptible species, we predicted that the decrease would not be quite as dramatic. So what did we find? Compared to the single species community of peepers, communities with peepers and toads had lower average number of cysts per community. Compared to the single wood frog only community, we found a significant decrease by 45% when toads were added to the community. In our most diverse community, we found a significant decrease by 30% compared to peeper-only communities, and a non-significant decrease compared to the wood frog-only community. And so, all in all, let me put it all together in this final slide here. We found that sodium chloride exposure altered host susceptibility to parasites. And by altering host susceptibility to parasites, we suggest that this modified the biodiversity disease relationship. And so one caveat out of this research is we focused on a single pollutant, salt. And we know that several other pollutants and a number of other pollutants can do different things to different to patterns of susceptibility. And so in thinking about the generalizability of this study, I think the main take home is the original prediction. We know that this biodiversity disease relationship has been equivocal in the literature. We suggest that one factor contributing to this is pollutants in the water, which alter susceptibility of hosts, which can influence that relationship. Altogether, we think that this study underscores the importance of integrating this global change sort of perspective into these types of disease studies. So pollutants can influence the magnitude of the biodiversity disease relationship. So thus far, I've talked about the effects of secondary salinization at the simple level with between hosts and parasites. We've just added in a little bit more complexity talking about multi-host systems. In this third and final section of the ecological perspective, We'll talk about the acknowledging of the fact that these interactions can occur in abiotic conditions that vary from one location to the next, and that variation in abiotic conditions may either exacerbate or dampen the effect of salt. And so in this section, the question that I will ask is, how do shifts in abiotic conditions modify the effect of sodium chloride? When it comes to abiotic conditions, one way that one mechanism for how abiotic conditions in aquatic systems can be modified is through the input of leaf litter. We know that different plant species have leaf chemistries that vary, and when these leaves are deposited into aquatic ecosystems and they break down, they can also modify abiotic conditions. In particular, one major threat to wetland ecosystems are invasive species. And so we're interested in understanding, as plant communities shift from native to invasive plants, how does this influence the influx of leaf litter and the abiotic condition into aquatic ecosystems? And more specifically, how do shifts in abiotic conditions caused by invasive plant leaf litter affect susceptibility to salt? And along the same theme as we've been talking about, how does invasive leaf litter modify host-parasite interactions? This is work done by my PhD student, Dr. Devin Jacobo, in collaboration with Dr. George Meindl. This is part of a course-based undergraduate research experience. Students enrolled in a class have the opportunity to go through the entire process of research, and it's an initiative to make research experiences more equitable and accessible to more people. So what is happening in this experiment? The students went out and collected leaf litter from several different species. In white are the native species, in red are the invasive species, and we paired them according to where they were found in the wetland. They took the leaf litter, soaked it for 28 days, and removed the leaf litter and placed the leachates into these beakers. Also, in each of these beakers, there's a single embryo, either an embryo of leopard frog, this is native to our area, or an embryo of the African clawed frog, which is non-native to our area and invasive in some parts of the US. So again, the question here is asking, how does being raised in different abiotic conditions influence an amphibian's susceptibility to salt? So once these embryos reach the tadpole stage, what we did was we conducted a standard toxicity assay to measure their susceptibility to salt. Here on the data slide, on the x-axis, we have the plant species, native or invasive. On the y-axis, we have average time to death. So you may notice that I condensed all the native species into one group and all the invasive species into one group for the sake of time. And also our principal components analysis indicate that the native leaf litter were more similar to each other than to the invasive litter. So what did we find? For leopard frogs, we found that those leopard frogs raised in invasive plant litter were less tolerant to sodium chloride. In other words, something about being reared in the invasive leachates made the leopard frogs more susceptible to salt. In contrast, we found the opposite pattern for the African clawed frog. Those African clawed frogs reared in invasive plant litter were actually more tolerant to sodium chloride. And so this result stood out to us because it was consistent with the idea of invasive facilitating invasive invasional meltdown ideas, in that the invasive leachate benefited the non-native invasive amphibian, but harmed the native amphibian. And so looking ahead, the next iteration of the class wanted to dive deeper into what might be driving these patterns. And so the following year, the next class looked at a number of different water quality metrics. Today, I'll focus on just general hardness. I'm focusing on general hardness because first, we found that invasive leaf litter leachates had a higher general hardness compared to native leaf litter. And this stood out to us because we knew that leopard frogs have a general hardness preference that is lower compared to African clawed frogs. And so the class hypothesized that the reason that the invasive leachates harmed leopard frogs is because the conditions were outside of the preference zone for the leopard frogs, but within the preference zone for the African clawed frogs. So to test these hypotheses, what the class did was they conducted another experiment. But this time, instead of manipulating leaf litter leachates, they manipulated general hardness. So on the figure to your right, this is the manipulation of hardness, and what you can see is that in the second study, the results were consistent with the original findings, which is on your left there. This suggests that the patterns that we found, it's fruitful for us to continue pursuing general hardness as a potential mechanism for why leopard frogs were negatively affected by the leachates, but African clawed frogs were benefited by the leachates, the invasive leachates. But all in all, the main take home here with this study is that the toxicity of sodium chloride was influenced by shifts in abiotic conditions, underscoring the importance of having this ecosystem level perspective when making predictions or testing the effects of pollutants on organisms. Similarly, when we're looking at what these invasive leaf litter leachates do to host parasite interactions, we find a very similar take-home message in that first, what Devin is doing in this study is he is exposing toads to the native and invasive leachates. He then moves these toads to clean water and adds tremotoads. So I think to point out here is that we did the exposures in clean water to isolate the effect of the leachates on the amphibian, on the host. We also did parasite exposures, but for the sake of time, I didn't include it here, but would be happy to talk about it with anybody if they are interested. So what did Devin find? He found that for tadpoles exposed to invasive leachates, they were more susceptible to the parasite compared to those that were exposed to the native leaf litter. Again, underscoring the importance of taking an ecosystem level perspective when looking at disease dynamics. So with that, this brings me to the end of the first section of this talk, the ecological perspective. From here, what I plan on doing is transitioning to the evolutionary perspective. So talking a little bit more about some of our work on the evolution of tolerance to antibiotics and to pesticides. So in this section, as a reminder, the broad question that I'll be asking is how does selection by pollutants influence host-parasite interactions? And I've organized this part of the talk by the different components of the disease triangle. So how does selection by pollutants influence parasites, influence hosts, and then their interaction? Now, specifically in this first section, I'll talk about the effects of pharmaceuticals from the perspective of parasites. I'll talk about BD. BD is a fungal pathogen that affects amphibians. It has been implicated as a pathogen that has caused declines worldwide. And this work that I'll be talking about is done by my newest PhD student, Isa, in collaboration with Dr. Obed Hernandez-Gomez and Dr. Karen Sauer. So, as we discussed before, host-parasite interactions do not occur in isolation. For example, factors in natural ecosystems like other microbes in the environment can influence the success of parasites. More specifically, we know that many bacterial species can produce antifungal properties that can inhibit BD growth or can even cause mortality. Similarly, on hosts, there are often microbiota, bacteria living in the microbiota that can also produce these antifungal compounds that again can limit the success of BD in infecting its host. So addressing the question, how does selection by pollutants influence host-parasite interactions? I alluded to it before, but we're interested in the evolution of tolerance to antibiotics, such as in a scenario like this. We know that the evolution of tolerance can be costly. And we were curious to ask, if these bacteria are allocating resources to antibiotic tolerance, how might it affect the antifungal properties of that they're producing? And ultimately, how might this then affect the success or the ability for BD to infect its host? So, does evolving tolerance to antibiotics influence the antifungal properties of supernatants produced by the microbes? To address this question, the model system that ESA is working on is Pseudomonas aeruginosa. These species are found in nature, both in the water column, in the planktonic form, as well as in a biofilm form, so similar to what might be found in the microbiota of hosts. This is an image of the planktonic form, and Pseudomonas is a great system because we have the ability to grow Pseudomonas, a strain that is susceptible to antibiotics, and also a strain that has evolved tolerance to antibiotics. This strain in particular has evolved tolerance by increasing the number of efflux pumps that can to take antibiotics out of the system. In addition to the planktonic form, we can also grow Pseudomonas in biofilm form, both susceptible to antibiotics as well as tolerant to antibiotics. So using these 4 different strains, what we can do is we can grow the bacteria susceptible to antibiotics, tolerant to antibiotics in the 2 forms, collect the supernatant. The supernatant is where the antifungal properties are found. And what we did was we generated 7 different concentrations of the supernatant, grew BD, JEL270 is the strain that we used, and what we measured was the proportion inhibition or the proportion enhancement of BD in the presence of these different supernatants. So moving on to the results. On the x-axis here, this is the supernatant concentration. On the y-axis is BD inhibition or enhancement. Anything above shows the supernatant enhancing BD growth. Anything below shows the supernatant inhibiting BD growth. And on this slide, this will just represent data from the planktonic form. So when reared in the presence of supernatant from bacteria that are susceptible to antibiotics, we found that all concentrations of the supernatant inhibited BD growth. Now, we predicted that bacteria that have evolved tolerance to antibiotics would face a cost. We predicted that their supernatant wouldn't be as strong as the susceptible group in terms of antifungal properties. When we looked at the data, we found that not only were these supernatants not inhibiting BD growth, but they actually cause an enhancement of BD growth in at least the lower concentrations that we used. In this slide, I won't spend too much time here other than to show that the results for the biofilm form of the bacteria are consistent with those of the planktonic, suggesting that there is some repeatability across different forms of the bacteria. So all in all, again, we were interested, does evolving tolerance to antibiotics influence the antifungal properties of Pseudomonas supernatant? We found that in both planktonic and biofilm forms, the supernatant of antibiotic susceptible bacteria inhibited BD growth, but antibiotic tolerant bacteria enhanced BD growth, suggesting that there may be some sort of cost associated with evolving tolerance to antibiotics. So where do we go from here? This is some of our relatively newest experiments. What ESA is working on now in terms of trying to figure out how generalizable this is in terms of conservation. She's looking at other strains of the BD. What initial studies have shown is perhaps that the patterns that I just talked about are strain specific. What she's hoping to do soon, as soon as our wood frog tadpoles are fully developed, she's going to be looking at how the supernatant direct exposure to the host influences the microbiota, the skin microbiota of the host, and then how this influences their susceptibility to the BD, since the microbiota serves as one of the first defenses against disease. So to conclude this section, evolutionary processes initiated by human activities in this part, pharmaceuticals like antibiotics, can influence parasites. In this case, antibiotics cause the evolution of tolerance in bacteria, which influence the antifungal properties and affected inhibition and enhancement of BD. In this next section, I'll switch gears from looking at the perspective of the parasite to thinking about the perspective of the host. And here we'll talk specifically about our work with pesticides. So how does selection by pesticides influence disease outcome? When it comes to the evolution of pesticide tolerance, generally what comes to mind first is this traditional paradigm of evolved tolerance. Genetic changes and constitutive traits due to natural selection. You have a population, it gets exposed to pollutants. There are some individuals that have a constitutive ability to express tolerance. If this trait is heritable, over time you get the evolution of constitutive tolerance. And so while this process allows populations to persist in the face of pollutants, there are a few limitations. First, it depends on standing genetic variation. You have to have those individuals that had that trait to begin with. And also, this process can take multiple generations. And this is important because human activities can cause rapid change. And the ability to evolve tolerance over time not happening fast enough to protect populations from a contamination event, like when pesticides are flowing into a community. And so we started to think about alternative paradigms for tolerance to pesticides. And this led us to phenotypic plasticity, or the capacity of a single genotype to exhibit variable phenotypes in different environments. For example, in the presence of a non-lethal predator cube, here are two tadpoles. They're from the same egg mass, they're the same species. The only difference is the tadpole on the bottom was raised in an environment with predators, which caused it to induce a deeper tail and a bright red tail, which allows it to better survive in a predator environment. Importantly, this induction of this trait happens within the lifetime of the tadpole, so it's a very rapid response. And so if these tadpoles can induce tolerance to a predator cue, we were curious, well, why not pesticide cues? Is it possible that tadpoles can induce tolerance to pesticides? And so we can turn to theory to make predictions about when we might expect plasticity, when we might expect constitutive tolerance. And so before I talk a little bit about the theory, I wanted to orient everyone to a reaction norm figure. On the x-axis, we have two environments, no pesticide or pesticide. On the y-axis, we have the trait of interest. In this case, it's pesticide tolerance. So if this tadpole were plastic to pesticide, we would see a reaction norm line that looks something like this. In environments with no pesticides, the tadpole would exhibit low tolerance. There's no need to be tolerant if pesticides are not present. In contrast, this same tadpole, if it has the ability to be plastic in the presence of pesticide, would have increased tolerance. They would have the ability to induce increased tolerance within its lifetime. Now, theory predicts that plasticity is favored in environments that are variable. So it makes sense if the environments are shifting, it makes sense to maintain the machinery to detect and to respond adaptively to those changing environments. An example in nature of variable pesticide environments might be a forested pond where for the most of the time there wouldn't be pesticides, but every once in a while perhaps there's an influx of pollutants like pesticides. In contrast, theory predicts that in consistent environments, plasticity is selected against since plasticity is costly. And so in consistent environments like ponds next to agriculture where the pesticides are always present, we might see genetic assimilation theory would predict that over time, we would get the loss of plasticity for constitutive tolerance. So tolerance that is always expressed regardless of whether the pesticide is in the environment or not. And this makes sense in the presence of consistent pesticide environments because why maintain the costly machinery if the pesticide is always there? So to test these predictions, we went out to northwestern Pennsylvania to 15 different populations of wood frogs that vary in their distance to agriculture and exposure to pollutants. Some variable, some consistent. And what we did was we exposed tadpoles to two environments, pesticide or no pesticide, and then we measured their tolerance. And so we were looking for plasticity, the sloped reaction norm line, or constitutive tolerance. So looking at the results, what we found, can tadpoles induce tolerance to pesticides? In this first panel, you'll see several populations that have that sloped reaction norm line. The solid line represents the average of these populations. These 5 populations are those that are far from agriculture, experiencing more variable pesticide environments. As we move closer and closer to agriculture, where populations are being exposed to more consistent pesticide environments, we're seeing this shift from a plastic slope reaction norm line to a flat constitutive line. And so a main take home from this study is that the evidence suggests that this loss of plasticity is consistent with genetic assimilation theory. Now, the main thing and why we're so excited about this particular finding is that it provides a really good model system for asking questions like this. How does selection by pesticides influence disease outcome? Because now we have characterized some populations that use plasticity as a strategy for tolerance, and you have other populations that use constitutive tolerance as a strategy. We know that these different strategies vary in cost, and so we can then ask an eco-evo question: does having inducible versus constitutive tolerance influence post-parasite outcomes? This is work done by Dr. Vanessa Werthner, who went back out to 15 populations of wood frogs. And what she did was she exposed each of these populations to either trematodes or ranavirus. Ranavirus is an emerging infectious disease. It's highly lethal. If it's in a system, it can cause mortality of that entire population within 2 weeks. So it's something of concern in the amphibian community. So what did Vanessa find? She, on this figure, on the x-axis, it's a range from inducible to constitutive tolerance. On the y-axis is trematode load. She found that populations with constitutive tolerance are less susceptible to trematodes. But the important point here is that your susceptibility to trematodes depends on whether you're expressing inducible or constitutive tolerance. Evolutionary mechanism to tolerance matters in disease susceptibility. What about ranavirus? We actually found the opposite pattern, where populations with constitutive tolerance were more susceptible to ranavirus. But again, the main point is evolutionary mechanism in terms of tolerance to pesticides influence susceptibility to disease. Now, I'll get back to that point, but a quick caveat: why is it that we're finding these different patterns associated with the different parasites? We're currently looking into this. The short answer is we don't quite know yet, but here is how we're trying to figure it out. We're turning to physiology to see if the populations vary in things like their corticosteroid level, stress, oxidative stress metrics. We're also looking at potential variation in microbiota diversity. Again, the microbiota for amphibians is really important because it serves as the first defense against disease. So does the microbiota differ between these different populations? Our intention is to, we're sort of at the phenomenological stage right now of this research, trying to pinpoint what might be driving these patterns, and eventually we hope to transition to the manipulative studies stage once we pinpoint factors that might stand out more. But so that's where we're going with this. But taking a step back, the take home again here is tolerance strategy influences susceptibility to disease. You have tadpoles that exhibit tolerance via plasticity, inducible tolerance, and via constitutive means. And this influences their susceptibility to disease. And for us, this is foundational because it forms sort of the base of a lot of our future work. In the future, as I mentioned before, we're really interested in this idea of biodiversity disease relationships. In this past research that I showed earlier, we showed that pollutant exposure altered host susceptibility to parasites. And this shift in host susceptibility is that one factor that could be modifying this biodiversity disease relationship. In this past work, I just showed that we have tadpoles exhibiting inducible versus constitutive tolerance, and these different evolutionary mechanisms influence disease susceptibility. And so looking ahead, what we're hoping to dive into is this question: does pesticide tolerance strategy, is it a cryptic factor that can influence the biodiversity disease relationship? So how we can integrate in that evolutionary perspective again into this biodiversity disease relationships type of work. We intend to do this using manipulative studies similar to before. We'll manipulate biodiversity in the lab. But of course, a major limitation of these manipulative studies is it doesn't really incorporate a lot of the complexity that we find in nature. And so we also hope to run field studies to look at how human activities can influence the biodiversity disease relationship. And with this, a major challenge is just the sheer amount of work it takes to also do field studies. And so to address this, we hope to involve community scientists to help us with our field work. And when it comes to community or citizen science, an important consideration is how do we ensure that the quality of the work is high. And so, as I promised before, I'll spend just 5 minutes talking here about how we plan to use art as a way to facilitate science communication to improve the quality of the data collected by our citizen scientists on this project. So specifically, we had to develop a way of asking, a way of improving public understanding of wetland concepts. To do this, we focused on third graders. We're interested in third graders because studies show that this is an important age to get kids to care about natural resources, but also this care is retained into adulthood. And so can art improve public understanding of wetland concepts? What wetland concepts did we want to focus on? Our intention was we wanted to choose something that third graders may not necessarily care about. And so for us, that meant trematodes. Obviously, we care a ton about trematodes. It's what we work with, but we didn't think that the third graders would initially care. So we used art to generate resources to teach kids about parasites. And the resources we thought that would work would be first a children's book. I'm showing the progression. So this is my art of it. I obviously am not an artist, but this is my idea of the story. Ben McLaughlin, an undergraduate in my lab, translated this into the book that it is today. This follows Gemma Jones as she meets Talia the trematode. They learn about the different complex life cycle of the parasite as it enters into the different hosts. It's paired with the lesson plan. And so we use art to generate this resource and use these resources to then train educators. We taught teachers how to teach students about wetland concepts, including parasites. And then once the teachers taught the content, their students were bused to a local museum where they had hands-on activities where they met different members of the book, like the amphibians, the parasites. And then we also were able to assess the effectiveness of conveying these wetland concepts. Here I'm just showing the parasite questions because I thought that these were the most interesting for the students. We asked them to identify a parasite. We asked them, are parasites bad? This actually came about because unfortunately we had a principal pull an entire class out of the program because the principal was worried that parents would be upset because parasites are bad. So we wanted to dig a little bit deeper into this idea. And then are parasites important? So in all 3 questions, we see a significant increase, which is really encouraging that we could use this sort of 3-step format to effectively convey information to third graders. And so back to this portion, how do we train students, third graders, to collect accurate data? Where we are now, this is the work of Kira Ricky. She is a PhD student in my lab that is focused on science communication, so she'll be getting her PhD in science communication. She worked with undergraduates to generate content again. We've generated a graphic novel, and this is intended to teach third graders about biodiversity. This is the undergraduate team that developed the graphic novel. And it's paired with a traditional lesson, so a PowerPoint. And so the Kira taught these lessons to teachers in a teacher workshop. The teachers are split in between these two lessons. They will then they're third graders, and then we'll have an opportunity to either look at the perception of science and also measure science content. And then from there, we hope that the students will be trained well enough to collect biodiversity disease data, and they'll be able to contribute to something that looks like this, measuring biodiversity in our nature preserve, and then also linking that to biodiversity, to disease, and looking at how human activities influence this relationship. So with that, a quick summary, we are interested in pollutants and how they influence host-parasite interactions. We look at it from the ecological perspective at different organization levels, from the individual, physiological, community, and ecosystem level. We're interested in evolutionary perspective, how selection influences host-parasite interactions. And science communication and engagement is something that is really important to us, and we do it at multiple levels. And none of this would be possible without our funding sources and also the amazing students that we have in the lab. This is just some of them. And they do all the awesome work. I just have the privilege of presenting it. And with that, I wanted to make a quick pitch. My lab is actually moving to University of Wisconsin-Madison, and we're hoping to recruit. If there is anybody interested in the work that I presented today, which is why I presented it the way that I did as an overview, there's a QR code to scan for details. Please reach out and apply if you are interested. So with that, thank you so much for your attention. I know that I presented a lot. I'd be happy to take any questions at this time if there are any.
Yeah, thanks, Dr. Baugh. I think everyone can sort of shout out questions if they have them, or if you'd rather put it in the chat, I'll facilitate the chat and pass those questions on. Really excellent talk, though. It was really fantastic, so thanks.
Appreciate it.
Hello, can I ask a question?
Yep.
Hi, this is Tim. I just wanted to say a very cool presentation. As a previous student who learned about this project in your class, it was really cool to see it being presented as it is right now. So it was really cool to be here. So I was very curious because you'd mentioned about looking into the abiotic factors such as pesticides in the host parasite interactions in this project. A lot of my work recently has been about eutrophication and how the influx of nitrogen and phosphorus impact harmful algal blooms. So I was wondering, have you ever considered, or how would you think the influx of nitrogen and phosphorus would impact the host parasite interactions with your tadpoles and trematodes?
I think that's an excellent question. It's good to hear from you, Tim. So to answer your question, actually, we have done that study. We were interested in microcystins. And what Nick and a few undergrads did is they looked at how exposure to microcystins, which are toxic chemicals produced when there are algal blooms, influence susceptibility of host to disease. And we found that at environmentally relevant concentrations of these microcystins, we see an increase in susceptibility to the trematode. We've only had a chance to test it on the trematode. We're recently just getting back into the BD and the virus work, so we don't know what the microcystins do to them just yet. But yes, we think it's a very important pollutant.
Very cool. Thank you very much.
Mm-hmm.
It was great to see you too.
Hi, I have a question as well. First, I wanted to say, awesome talk. I used to do a lot of parasitology, trematode work back in the day, so I love hearing about it again. I thought your work on the salinity impacting the infected part of this area to be very interesting. and it made me wonder, did you look at how the salinity might impact the snail intermediate host and how that might influence the increased production of Cercaria, or possibly even, I imagine it would negatively harm the snail host, which would probably negatively impact the ability to produce Cercaria. So there could be that's my initial thought. And if you could talk about that at all, I'd love to hear it.
Yeah, that is an excellent question in terms of the complexity of this system. So the snails are producing the Cercaria, which I didn't talk about in the complex life cycle. So the snails are producing the Cercaria that then infect the host, the tadpole host. So what we have found, and we haven't done this with salt, but we have done it with pesticides. Snails are tolerant of everything that we have thrown at them. So it doesn't seem to matter to the snail at all in terms of their abundance, their size. What we haven't looked at is how it affects production of Cercaria, or even the number of radiae in the snail. And the reason why we haven't done this is that it is extremely difficult to get to that snail stage. So you have to raise chicks, the final host, get adults from the host, have eggs from the host of the terminal host, the avian host, and then use that to infect a snail. And so that part of the life cycle we have gotten to with the pesticides, but not salt just yet.
Botham, do you have a question?
Yes, hi. Great talk, Jessica. As a follow-up to Chris's question, I was wondering the exposure, the host parasite exposure you got with the salt, was that exposure done under salty environment or unsalty environment? In the former, if it's salty, what is the effect of the salt on the parasite itself? If the other way around, what is the effect of the lack of salt on a host that was raised under salty environment? So a rapid change in salinity. I'm just curious how you did it.
Yes, I think I'm understanding your question correctly, but if I misanswer, please ask the follow up. So how we did it was we had tadpole exposure to salt, and they were moved to clean water. So we isolated the effects of salt on the host, and then allowed the parasites to infect the host. And then we also had, which I didn't present today, we had a time to death on Cercaria. So we took the parasite and exposed them to salt and just measured how long it took for them to be immobile. And then a third section was we exposed them together so that we could isolate each part of it. But for the sake of time, I didn't include those in the talk.
Great, thank you.
Mm-hmm.
So we are at time, but just to ask one final question, just because I think you prompted it when you presented, were there any effects of the leachates on the parasites themselves?
Yes, there were actually. When it came to the invasive leachates, the invasive leachates actually caused a decrease in the number of Cercaria. So the Cercaria that were exposed to the invasive leachates, they became immobile faster. And so what is interesting and what we hope to ask next is it abundance that matters of those that survive and can still be mobile? Can they still effectively infect the host? And from there, what is the survival of the metacercaria? And so that's one thing. We don't know the state of the metacercaria after they have infected the host. Like, are they viable? How long are they viable? Lots of questions. It seems like we answered one and 100 more, which is the coolest thing about, I think, what we get to do.
Yeah, thanks. Does anyone have any final questions? I know we're at time here.
I have another question if I'm allowed, and no one else is jumping at it. I was curious, so the infective rate of the parasites infecting the larval frogs, tadpoles. Sorry, lost the terminology. I saw it was about, you add 50 trematodes and you get about 12 to 16, somewhere around that range. What are the host parameters that actually control infectivity? Do you know that? Like what parts of the innate or adaptive immune system are regulating the infective load?
So a lot of it actually is behavioral responses. So these tadpoles will, they have a tail flicks that will flick away the parasite. They'll have like bursts of speed that can, like when they detect it, they'll get away from it. But when the parasites get into the kidneys is where they insist. We do know that they make it so that it's easier for other parasites to infect. And something that is really cool about this system is that there are two kidneys, so oftentimes there's a left kidney bias, so that the trematodes will all go into one side, and it's adaptive because they can't, they don't want the host to die. They need the host to be consumed so that it can move to the next part of its life cycle. So a really, really cool system, complex in many ways to answer questions, but I think there is a wide variety of different mechanisms in terms of tolerance from the host perspective.
That's awesome. Thank you very much. I always love hearing about the behavioral stuff impacting parasitology.
Yeah, this talk was for you, Chris.
I had a feeling.
All right, so we're a little past time here. I think most people have had to sign off, but thank you so much again for talking and giving such a great presentation. Yeah, and I hope that we can keep in touch, and I'm sure other people from SoMAS will reach out to you as well. So.
Appreciate the invite, Kim. It's awesome to see you again.
Yeah,
we'll talk again soon.
Sounds great. All right, talk to you later. Bye.
Bye-bye.